So, after initial topics with chemotherapy, we are going for the next topic. That is, we have seen earlier what are various general considerations in chemotherapy, what are various terminology, what we can combine, what we cannot, what are various reasons for that. Then next is, we have seen the first antimicrobial agent which was found, that was sulfonamide, and various combinations, their pharmacokinetic details, classification, etc. we have seen. The next topic in that is penicillins. They are very popular. Again, they were the first antibiotics which were introduced. If you remember, I use a word that is, we call it as a serendipity. It was found by just by chance, and then the scientist, that is Alexander Fleming, he went for more studies. He confirmed that there is something which was there, but he could not isolate. Then there were other scientists who were named as a Fleury and Chain, who got Nobel. All these three got Nobel. Fleming also, then Fleury and Chain also, all these got Nobel Prize. So that these two got for separation of this molecule and finding out the structure, what it is. So this is how penicillin came into structure. These are actually the agents to what we call as a agents, because of the chemical structure we call them as a beta-lactam antibiotics. And second thing, they are acting on the cell wall synthesis. They inhibit the cell wall synthesis, and thereby they affect the bacterial growth, that is bacterial, they affect the bacterial what we can say multiplication. And this is how these agents are there. They are very still out of that, the second one, cephalosporin, are very, very common. Even carbapenems and monobactams are being used nowadays. Let us have a brief classification of this, which should help us to understand it in a better way. So we are classifying them into one class, to what we call it as a inhibitor of cell wall synthesis. So we have got so many agents under this class, we call them as an inhibitor of cell wall synthesis. In that we have got two subclasses. One are, we call them as a beta-lactam antibiotics. Why we call them as a beta-lactam? Because chemically they have a structure where there is one beta-lactam ring is present, and because of that beta-lactam ring we call them as a beta-lactam antibiotics. Then the another one, the group, we call them as a other antibiotics, because they are not classified under any such class, and they are separate, they are isolated, they don't have any big group of compounds into it. So out of that, these are vancomycin and bacitracin. These are two which we classify as a other. Now you can find the biggest one, the major one is, we call them as a penicillins. Then second major one is called as a cephalosporin. Third one are the carbapenems, and then monobactams. These are the four most important, or one of the most, rather I can say, one of the most important groups of antibiotics, and which are still used, still widely used. Some of these are still widely used, and they are very, very effective in control of various microorganisms. Now let us see one by one. We'll start with penicillin, what they are. So first one in that is penicillin. So it was the first antibiotic, as I told you, it is the first antibiotic which was used clinically. I told you that Alexander Fleming, who accidentally or by chance luckily discovered that because of the contamination of fungus into microbial plate, if you have done microbiology and you have seen the Petri plate, because of that fungal growth or fungal portion which got into that plate, there was a zone of inhibition, and then they could not do it. Yeah. Hello. Just a minute. I'll Sorry, it was disconnected. Okay, just a minute, please hold on. So we were discussing that is penicillin was the first antibiotic which was to be used clinically. Alexander Fleming accidentally found it. He was fortunate, he could observe it, and then he was not sure. Then whatsoever his reports were there that were studied, and as I told you, chain and flurry, or people call it as a flurry and chain. They separated it from the cultures of Penicillium notatum. It was a fungus, and presently it is obtained from the cultures of Penicillium chrysogenum. So this is the basic brief history of penicillin. This penicillin is having a typical structure. We call it as a two rings are there. One is basic ring, that is we call it as a thiazolidine ring, which is fused to another ring, to what we call it as a beta-lactam ring, and to this beta-lactam ring there are some various side chains are there. Now this is again you can study this in medicinal chemistry. We call it as a structure activity relationship. So what are the impact of various substitutions at various places, and accordingly we can get variety of molecules. So this is what we call it as a structure activity relationship. Then next in that is what was the first, that was called as a natural penicillin. So that was first one was, that was a natural penicillin. It was also called, or it is also called as a penicillin G, and the R, that is side chain, that is a group's positions, it is known as a benzyl side chain. But in semi-synthetic penicillin, R is anything other than benzyl moiety. So the first one was called as a benzyl penicillin, or penicillin G, or natural penicillin. That is one and the same. Okay. So these are the Shivani Bagure, something is problem. So it is penicillin structures features. If we see, this is the ring, this is the this is what we call it as a thiazolidine ring. This is the next ring, we call it as a beta-lactam. This is very fixed ring, and this is the place where this various enzymes, that is we call it as a beta-lactamase or penicillinase, that enzyme which will be breaking this, and thus this beta-lactam chain will be breaking. You can see the blue color one, penicillinase. So this structure, overall this structure is called as a penicillin when the ring A and ring B is present, and then the various groups are attached to what we call it as a R. So these substituents are there, and on that basis of these substituents, these particular compounds, they are having variety of chemicals, variety of basic what we can say congeners or similar agents of this class. So moiety A and B is together consider 6 amino penicillinamic acid nucleus. So this moiety A and B, okay, this together up to this is called as a 6 amino penicillinamic acid, and this is very, or it is briefly it is abbreviated as 6 APA. So once this 6 APA is formed, then the other molecules are synthesized, and if we use this nucleus, if we use this 6 amino penicillinic acid as a nucleus, then we get the other agent to what we call as a semi-synthetic. So on that basis we will have the classification, we can call it as a natural, example is penicillin G, penicillin V. Then the second one are the semi-synthetic, means half portion is natural, half portion is synthetic. What is natural? Then 6 amino penicillinic acid is natural, and in that group the other groups are attached externally. After that, then we call it as a semi-synthetic, and synthetic means completely outside. There is no need of bacteria or culture, nothing will come from bacteria, we call it as a synthetic. So this penicillin nuclease is a chief structural requirement for the antibacterial activity, and the side chain will determine various antibacterial and pharmacological characteristics of the particular type of penicillin. So this is how we have a variety of structures. So we have got a variety of natural penicillin, penicillin F, penicillin G, penicillin X, penicillin penicillin K. Penicillin G is a benzyl penicillin. Its sodium and potassium salt are more there. It has got greatest antibacterial activity. This is very important. This has got greatest antibacterial activity. It is only natural penicillin which is used clinically nowadays, that is penicillin G. Only unfortunately it has got very narrow spectrum. We shall see it. Okay. So penicillin is given in the form of units. It is not given in the milligrams or microgram doses. So how these units are calculated? The activity of natural penicillin is described in unit, but the other analogues like semi-synthetic and synthetic analogues, they are given in their weight form. That is, okay, I'll just check. I'll check because it was cut off in between. Oh. क्लास होऊन जाऊ दे बेटा माझं. क्लास आला आहे ना, माझं काम झालं तुझं. छान, चलो ऑल द बेस्ट. काही कोणाला मागवायची गरज नाही. हां झालं तुझं बघून घे ना. मला वाटतं जर जास्त चांगलं काम झालं. तुझ्या डगचं काय झालं? एक मधून रिमाइंड करत द्यायचं घ्यायचं कृपया. हां ठीक आहे ना, ठीक आहे, रिपोर्ट करत द्यायचं. चालेल. I'll just go back for this penicillin. So what it has got is the basic unit is got two things, that is thiazolidine ring and beta-lactam ring. Then the natural penicillin, penicillin G, R side chains is a benzyl side chain. In semi-synthetic, it is R is anything other than benzyl moiety that we have seen. This is chemical structure of penicillin. There are A and B, these are two rings, and this A and B ring together, it makes, we call it as a 6-aminopenicillinic acid. Then the substitutes are there which are attached here at various places. You can see this blue color lines are there. There are two big lines are there. We can see almost fluorescent dark blue. So this is one is that is called as amidase, which are this enzyme will break this side chain, and thus it will make inactive. And another is most important is we call it as a penicillinase or beta-lactamase. These enzymes will break down this penicillin ring, and therefore they will make them inactive. This structure together, as I told you, it is called as a 6-aminopenicillinic acid, and this into this, when synthetically we attach some structure, we call it as a semi-synthetic one. So we have got various synthetic and semi-synthetic agents which are available, and penicillin nuclein is a chief structural requirement for antibacterial activity. Only we change the side chain when particularly it is semi-synthetic one. There are many natural penicillins are available: penicillin F, penicillin G, penicillin X, penicillin K, and penicillin G is the having highest grade or antimicrobial activity. I told you that it has got only problem is limitation is it has got few difficulties. We shall see it, and because of that, despite of that, it is not much widely used nowadays. Then next is penicillin, which is natural. It was not given in a milligram, microgram, picogram format. It is given in the format of units, just like we give it in insulin or some other such agents, where we give it in the activity in the form of unit. So it was activity was described in unit, and how it is there, that is crystalline sodium penicillin G contains 1600 units per milligram. 1 unit of penicillin G is equal to 0.6 microgram, or 600 milligram of penicillin is equal to 1 million units. So on that basis it was always given, that is, and semi-synthetic penicillin are prescribed by weight. I told you 500 milligram or 250 or 400 milligram accordingly, depending on the type of this semi-synthetic, they are prescribed by weight basis, while the natural penicillin is prescribed on the unit basis. Now for that, let me tell you story. Whatsoever these natural compounds were there, maybe insulin, penicillin, since they were derived from natural sources, it was not very sure that what compound it was. At that time purification techniques, then assay techniques were not big, so they were based on the activities, that is how much is able to kill the microorganism, or how much insulin is able to bring down the glucose down. So since they were natural extracts or naturally they were obtained, so in that cases the most important thing was activity. And so batch to batch variation, instead of milligram, it was done on the basis of the activity. So if it was seen that all the, when the batch is passing, the activity is same, okay, and on that basis the units were made. So these units were, in earlier days it was made, it has continued in case of penicillin G and insulin, but this was the reason. Even though nowadays we have so many methods of purification, we have so many methods of confirmation, assay methods, etc., but this is the reason why we use unit methods for these natural compounds. So 1 milligram is equal to 1600 unit, or or vice versa, and therefore we can calculate it accordingly. 0.6 Ug is equal to 1 unit, 6 is equal to this 10 units, 6000 is equal to 10,000 units, and accordingly it is there. Okay. So this is how we are calculating. What is the mechanism of action? These agents are interfering with the synthesis of bacterial cell and cell wall, and because of that they are able to destroy the cell wall, and thus it will make the bacteria vulnerable for the death. The bacteria are always surrounded by a thick wall of that gives stability and rigidity to their cell structure. Cell wall is compound of the typical peptide chains and glycan chain. We call them it as a peptidoglycan chain. It is extensively cross-linked, means it is a tiles-like structure. If you might have seen various tiles at various places. So how the tiles, if you see the lines in between, there are cross-linking are there. Peptidoglycan layer envelopes the cell and does not allow bacteria to swell and prevent death due to lysis. So it will not allow bacteria to get died because of the lysis of the bacteria. But when this is not there, then bacteria can be easily lysed. So how this happens? There are two important glycans are there in this. There are two amino sugars are present in this glycan one. That is called as a N-acetylmuramic acid, that is called as a NAM, we can say, and another one is known as a N-acetylglucosamine, NAG. So this NAG and NAM, these are two important glycan chains which are there to the amino sugars or to the protein moieties, and there is a pentapeptide side chain is linked to NAM sugar and it forms the peptidoglycan molecule. These peptidoglycan residues are linked together, and the UDP, that is uridyl diphosphate, is split off, and then this makes a big molecule of this peptidoglycan. So final step in that is a cleavage of D-terminal aniline of the peptide trans by transpeptidase. And because of, I think we have seen last time when we have seen mechanism of action, there are protein binding proteins are there in earlier type of presentation we have made it, that is there are some protein binding penicillin binding proteins, I'm sorry, this this PBPs means penicillin binding proteins are there. They are there with the transmembrane surface, and there are some enzymes are also present to what we call as a penicillinase or beta-lactamase. The energy is released which is utilized for establishing cross-linkage between peptide chains of the neighboring strands, and this cross-linking provides stability and rigidity to cell wall. This is what natural activity. Now what penicillins they are doing is they inhibit the bacterial cell wall synthesis. How they inhibit? They inhibit the penicillin binding proteins which are catalyzed process of transpeptidation, and because of that, instead of normal substrates, they will get inserted into this cross linking. They will maintain the close knit structure of the bacterial cell wall, which does not occur, and this last step in the peptidoglycan synthesis will be affected by these penicillins. When susceptible bacteria divide in presence of beta-lactam antibiotic, the cell wall deficient sperm are produced, and they are easily killed or easily get destroyed of that. The interior of bacterium is hyperosmotic, and so it will swell, it will take the water in. Because of the osmotic drive, this will take out, they will cause swelling of the cells and bursting of the cell, and because of the lysis of bacteria, these penicillin are bactericidal in nature. Okay. So this lytic effect may also be due to de-repression of the some of the bacterial autolysins. Autolysins are their own chemicals. So their rapid cell wall synthesis occur when organisms are actively multiplying. So during this rapid, when actively multiplying, at every time this penicillin will be taken with, and they will be every time the penicillin will be taken during their multiplication, and because of this penicillin will be incorporated, and thus it will become a bactericidal activity. So they are more lethal in this phase when there is a rapid development is going on. Now if you remember, during general consideration we have seen one thing particularly. One thing was what? That combination of various antibacterial agent. Now you can think about it. Now suppose we give any bacteriostatic agent along with penicillin, what will happen? Because penicillins are highly, strongly bactericidal agent, they are very lethal one, and they are acting at what pace? When bacteria are multiplying. At the time of multiplication, when next bacteria cell wall synthesis is going on, at that time they are going to be incorporated, and because of their incorporation into it, that will make a situation like this. So this particular thing is called as a what we can say, if we add any antibacterial agent which is affecting the protein synthesis, or that is it is affecting the ribosomes. So what it will do, ribosomes will be affected, because of that protein synthesis will not be there. If protein synthesis or DNA synthesis will not be there, new replication will not be there. So if new replication is not there, that means no further cell division will be there. This is what bacteriostatics are doing. But beta-lactams are effective not in the current situation of the bacteria. They are effective when the bacteria is developing, bacteria is dividing, and at that time when the bacteria is taking up penicillin in formation of cell wall. I hope you are understanding. So this is how, why we should not, or why the combination will be affecting. This is the explanation which was earlier told, that in a very first class of chemotherapy I told you about this, that what kind of combinations can be done. So if the major action is bactericidal, then we should not combine bacteriostatic with it. Peptidoglycan cell wall is unique to bacteria. It is not at all present in human beings, and no such substance is synthesized by any higher animal, and because of that penicillins are practically non-toxic to man. When general pharmacology teaching, I used to tell that if we calculate therapeutic index, it may be in hundreds or thousands. Any type of dose of penicillin can be given to person, there is no problem, except we shall see what are the exceptions. The biggest thing is they are particularly more active in gram-positive bacteria. Particularly natural penicillins are more active, because cell wall is entirely made up of peptidoglycan in gram-positive. It is around 50 to 100 layer thick. It is extensively cross-linked, and it is may be considered as a single giant peptide molecule, means totally what we can say, just like a structure is made, suppose in building or somewhere we make a structure, total structure from all sides. So it is like a structure which is made from all sides, and that structure is very prominent structure. It is a sort of skeleton of the bacteria. This peptidoglycan layer is easily accessible to beta-lactam antibiotic, and inhibition of transpeptidation reaction, that becomes inhibition easier because of that in gram-positive. Gram negative has got two membranes, outer membrane and inner membrane. Peptidoglycan is sandwiched between these two. Okay, and because of that, the outer membrane, which is consist of lipopolysaccharides, they are having narrow porine channels. So these are pore-like channels are there, small porine channels are there. Now these are making they are so small that penicillin cannot go into it, or beta lactams cannot cross through it. They are therefore they are having weaker activity against gram negative bacteria, and therefore penicillin G is highest susceptibility towards gram positive and almost no or very little towards gram negative. So this is very important. Now simultaneously we are getting explanation of their spectrum of action. If you remember, we call it as a spectrum of action, that against how many kinds of different type of bacteria they can be active. So here it is, penicillin G is a narrow spectrum antibiotic because it can act only against gram positive and it is having very little or less activity towards gram negative. So this is how the gram positive structures are there. There is a cytoplasmic membrane, peptidoglycan cell wall, and in that it can be incorporated. Now if we see the gram negative one, then this is in between these two structures, outer membrane, inner membrane, and there is a peptidoglycan in between, blue colored one, you can see this. So this blue colored structure, that shows that it is a peptidoglycan, and because of that the penetration will not be possible. These are the channels, or porine structure is there, we can see this, and this porine structure will not allow the penicillin, because they are very small in size and will not allow penicillin molecule to cross through. So penicillin with hydrophilic characters like ampicillin and amoxicillin, they are hydrophilic in nature, they can diffuse through the porine channels, and therefore they can show activity against some gram negative bacteria also, but they cannot show any action against Pseudomonas aeruginosa. This is another kind of bacteria where these bacteria lack the classical permeable porine channels, so it will not be active against Pseudomonas. So let us see mechanism in brief, how they are acting. Cell wall of bacteria is essential for normal growth and development. There is a peptidoglycan cell wall is there, which is highly cross-linked lattice work structure, and it is a continuous structure. The glycan chains, like linear strands for two amino sugars like NAG, NAM, that is NAG and NAM, they are cross-linked by the peptide chains, and they are provides a rigid mechanical stability. Particularly they do not allow the osmotic changes, and because of that the water molecules cannot go inside, and the hyperosmolarity which is there inside the bacterial cytoplasm, that will remain as such. But as soon as it is not there, then because of that the osmotic pressure wise water will go inside, and too much water will cause bursting of the cell. Now why there is a resistance to this, how the bacteria there, we have seen as a general mechanism, but let us see what. There are two ways we can say. There are inherent and acquired. Inherent means because of the normal life cycle of the or normal metabolic activities of the bacteria, they are at normal level only, they are able to prevent, they are able to we can say combat with the action of the antibacterial. And acquire means initially they were susceptible, but after some time they acquire them, there is a change in their genetic structure because of mutagenesis or any other such reasons, and they become they acquire this immunity. Example of this inherent is what? The gram negative bacteria, they are inherently resistant. Why? Because the presence of porine channel through which it cannot going, and because of the this peptidoglycan layer which is sandwiched, because of that there is a resistance. And further what there is a two thing is there, because of the deeper localization of the target enzymes and penicillin binding proteins, they will not be acting. There is a loss of alteration of the porine channels. This is another mechanism. Particularly suppose example we have seen that ampicillin, amoxicillin, they are active against gram negative bacteria, but then after some time somebody will say there is a resistance form. So first thing is there, that is deeper localization was there, which was natural, that is fine, but then there can be loss or alteration of the porine channels which are be there. So penicillin G is unable to penetrate into this, and another acquired are there, that is production of penicillin is acquired, and secondly change in the porine channels. that can also be acquired. These are various acquired, particularly when it was effective earlier. Example is ampicillin, amoxicillin. So penicillinase is the enzyme, or it is also called as a beta lactamase. It opens the beta lactam ring, and once the beta lactam ring is broken, then the penicillin G is inactivated. There are all variety of bacteria they can do it. Example of these are all Staphylococcus. Now if you have studied microbiology, you must find that there are many Staphylococcus bacteria are there. Example is Staphylococcus aureus, etc. Okay. Then Gonococcus, Bacillus subtilis, E. coli, and then Haemophila influenza, etc. There are many such agents which are available. Then they are producing penicillinase, and therefore they cannot be used. So there are two things which are there. Let us see what it is. Penicillinase are the enzymes. They are acquired in gram-positive organism. It was not there earlier. This can produce large quantity of penicillinase, which diffuse in the surroundings, and this protects the bacteria when there is a resistant one. In gram-negative bacteria, it becomes as a constitutive enzyme, although it is a small quantity, but it is strategically located between these lipoproteins and peptidoglycan. So it will not be there, and again because of this, it will not allow the action on this particular bacteria. So this is how resistance is formed. So this is the structures, that is there are PBPs, that is penicillin binding proteins, in gram-negative and gram-positive, and the enzymes are present. Porin is there in gram-negative, and these enzymes are working for structural synthesis of cell, that is peptidoglycans. Now in some places, why there is a resistance, that is particularly in gram-negative bacteria, there is a efflux pump. Efflux pump means as soon as it is taken up by the cell or near cell, before it do any metabolism, they are thrown out of the cell. The pumps are developed, pumps are made, that selectively penicillin is thrown out of the cell. We can say just like active transport mechanism, sodium potassium pump you have seen, like that pumps will be there, and these pumps will be doing the throwing out of the this penicillin. So first is penicillin G. It is a natural penicillin. It has got narrow spectrum. We have seen that it is very active against gram-positive, and it is primarily active against gram-positive bacteria, very little against gram-negative and anaerobic bacteria. Very wide spectrum it has got. Gram-positive cocci, streptococci are highly sensitive, like pneumococci, Staphylococcus aureus. Around only thing is which Staph aureus is around 90% has got acquired the resistance, and therefore penicillin nowadays has no effective. Now therefore if you know it, or you can keep it in mind, when we say that is pharmacoepidemiology, that is particularly we study about various kind of such antibiotics, where these antibiotics are in certain areas where because of the indiscriminate use, or maybe bacteria which are more present, more predominant, these are forming the resistance, and then in that area particularly there is no point in using these antimicrobial agents. So although it was earlier known, although maybe 5 years back, 10 years back, 2 years back, it was known that it was very good antibiotic for any infection, and now if it is known that resistance is formed by various tests, various research activities, then there is no point in prescribing that antimicrobial agent in that particular area, or when it is known. Therefore, if you remember in our first lecture, the introductory lecture or basic principle, at many places nowadays, many hospitals, they have their in-house microbiology unit. They go for culture testing, they check which kind of microorganism is there, they study the culture, and then next thing they do is the sensitivity testing, that is this culture, this microorganism which is there, which is sensitive to which kind of antimicrobial agent. So even though earlier 2 years back, 4 years back, in theory, in pharmacology book, maybe Goodman Gilman or whatsoever it may be, it is showing that it was active, it is having this kind of spectrum, but finally actually decision will be made on that basis of what kind of microorganism is there and whether it is sensitive or not. So the gram-positive bacillus are there, like Bacillus anthracis, then Clostridium diphtheria, all Clostridia, Listeria, they are highly sensitive. Few of these gram-negative cocci, they are susceptible. Example are N. gonorrhea, it was earlier susceptible, nowadays it is become resistant. N. meningitis are susceptible to penicillin G. Then there are some gram-negative bacilli are there. Example, Mycobacterium tuberculosis, Rickettsia, Chlamydia, Protozoa fungi. They are totally unresponsive. There is no actions on these bacilli like tuberculosis, Rickettsia, Chlamydia, Protozoa fungi, viruses. There is no point, nothing. It is just waste of time, money, and maybe the condition of the patient. Most anaerobic microorganisms, including Clostridium species, they are highly sensitive. Then some spirochetes are there, they are highly sensitive. These spirochetes, one of the spirochete which causes dandruff, there are many spirochetes which can cause a variety of infection. They are usually they do not cause, but once they cause, they are very difficult to treat. So what are various agents which are there? Strepto, that is streptococci, pneumococci, meningococci, anaerobic spirochetes, and in this type they are active because they are cocci are usually gram positive, so they are active against it and least active against other. Now what are the problem? When we say that penicillin G is the most active, it has got highest antibacterial activity, it is the most safest, then what are our problems? Problem is what? First is acid labile. It cannot be given by oral route at any time because it is destroyed by gastric acid, cannot be absorbed from other mucosal areas. It is absorbed only from intramuscular site, so always it has to be given as a intramuscular injection. It is absorption from intramuscular is rapid, it reaches in most of the body fluid, half life is 30 minutes. To prolong the duration of action, it is combined with some of the agents like we call it as a procaine, benzathine. These are the chemicals. They will act as a prodrug, and this procaine penicillin or benzathine penicillin, then slowly procaine and penicillin will be hydrolyzed. The penicillin will be active, procaine will be metabolized. But problem with that is procaine itself is a local anesthetic in nature, so it will cause its own adverse effects simultaneously. Is one thing. Second thing is done is how it is excreted. It is excreted by rapid renal excretion because 10% by this glomerular filtration and 90% by tubular secretion. So maximum excretion takes place by tubular secretion and it is always active. So this actively excreted in tubular in the last phase of urine formation. Recently, just in the initial days, we have studied diuretics. So in tubular secretion or in final tubules, we say this is acting, this is happening, and this tubular secretion we can block with the agent like probenecid. So this probenecid will block it. Probenecid will get excreted instead of penicillin. So when probenecid is given, penicillin will be means they will be compete for same site. Probenecid will be excreted, penicillin will go back into blood, and thus it will increase the plasma half-life. But it is rarely used for this purpose, probenecid, because again as you increase number of drug, it increases the adverse effect chances. Release penicillin slowly from the area in which they are injected and produce relatively low but persistent concentration of antibiotic in the blood can be maintained. This can be done. Various penicillins like penicillin G, that is called as sodium penicillin G, crystalline penicillin or benzyl penicillin, procaine penicillin G, benzathine penicillin, and fortified procaine penicillin G. These are various preparation. As I told you, all these are parenteral in nature. What are the therapeutic uses? They are for streptococcus, particularly drug of choice for sensitive strains, for pneumococcal. Again, for everywhere actually this word is very common. Sensitive strains, at most of the places they are replaced by the third generation of cephalosporin. Then meningococcal, gonorrhea, they are not reliable. It is not clear whether they will be able to do it. Syphilis, it is drug of choice. But what is the problem? We shall see about syphilis. Diphtheria, they are useful for the carriers who are there, or carrier means these are the persons who are not showing any sign symptom, but they are carrying the microorganism and they can transmit to other place. Tetanus and gas gangrene, these are again very severe from Clostridia tetani, and this, once it is there, it can be very dangerous, it can be lethal to the person. So in that it is useful. It is useful in the infection with the anaerobic, particularly dental infections, like mild to moderate infection at various places. In more severe infection, it be treated with 12 million to 20 million units of penicillin G. Gingivitis, then Vincentia angina, these are various places. Prophylactic in case of rheumatic fever, 1.2 million units intramuscular once a month, lifelong in a high risk people, because it reduces the immunity of the person. Bacterial means rheumatic fever reduces the immunity. Bacterial endocarditis, it is caused by dental excretion, endoscopies, catheterization, and in that cases it is used as a prophylactic in that. So in some of the other dental activities it is used. Then various prosthetic cardiac wall, that is prosthetic means artificial cardiac wall. Then congenital heart diseases like unpaired sinusoid CHD. In many conditions it can be there, wherever the walls are replaced and anything is there inserted in body, and then gram positive organism are expected that it will cause infection, they are used. Now second are semi-synthetic penicillin. We shall discuss the problems with all penicillin simultaneously. Semi-synthetic now we will take in next class. What are these semi-synthetic penicillin? We will classify them also, and what are various adverse effects of these which are common for that. We have already said one important thing is penicillin is the safest drug which is there, but at the same time it can be lethalist, it can be very dangerous in one situation. We shall see about it, how it is, and this will help us to understand basically what we call it as how these adverse effects are important, and what has to be done in that cases. So we shall see about all these drugs, various agents, various classification of these semi-synthetic agents or synthetic agents, and what are their importances. So I hope you are understanding this. If you have any difficulty, you can definitely put on our group so that we can try to resolve it, and take care. Be